Viruses are biological systems that do not have a cellular structure. Therefore, they do not have cells. Viruses exist in two forms: extracellular and cellular. Click on the glass to see the structure of a virion using the tobacco mosaic virus as an example. In extracellular form, viruses exist in the form of virions, particles that have a constant shape and a fairly simple structure. In this form, they cannot carry out the processes of self-reproduction or metabolism. The main purpose of this form of the virus is to find a new host cell and penetrate it. Click on the next glass to see the different forms of existence of viruses using HIV, the human immunodeficiency virus, as an example. In the cellular form of existence, viruses carry out the process of self-reproduction, using the resources and components of the host cell. At the end of the existence of this form, a large number of new virions accumulate in the cell, going out into the external environment and searching for other cells. Click on the cell to see how it happens. Viruses can reproduce only in living cells, as ribosomes, tRNA, ATP molecules, enzymes, and other components of cellular structures which are responsible for the synthesis of organic substances that viruses themselves do not have. Therefore, all viruses without exemption are intracellular parasites. They are not able to carry out the process of self-reproduction on their own. Viruses can be simple or complex. Simple viruses only contain nucleic acid and a protein shell, the capsid, which surrounds said acid. You've already considered a simple virus in our first example, a virion. Virions of complex viruses differ from simple ones thanks to the lipoprotein shell, very similar to cell membranes. Click on the next glass to see the structure of the complex virus, the coronavirus. It's not surprising that the envelope of the virus is similar to cell membranes, because this envelope is formed from the area of the cell membrane where it multiplies. It also includes specific proteins of the virus itself, which mainly perform a receptor function, recognize a damaged cell. Click to see the virus from the inside. Under the lipoprotein shell of the virion of a complex virus, there is a capsid, a shell consisting of proteins. Nucleic acid, which contains hereditary information, is located inside the capsid. Click on the next sample to see the herpes virus. Nucleic acid inside the capsid can be of only one type, RNA or DNA. Viruses, respectively, are divided into two large groups, RNA viruses and DNA viruses. The herpes virus is a DNA virus, and the coronavirus is an RNA virus. In addition to viruses, non-cellular forms of life also include viroids and prions. As mentioned before, viruses contain at least nucleic acid and proteins. Viroids and prions only have one of these two components, either nucleic acid or proteins. Click on the sample to learn more about viroids. Viroids are circular RNA molecules and do not contain any proteins. They are located in the nuclei or chloroplasts of plant cells and use their enzyme systems for their own reproduction. Viroids can be the causative agent of some plant diseases, such as in this potato. You're now observing a spindle tuber disease of potato tubers, the causative agent of which is PSTVD, potato spindle tuber viroid. Press Next to learn about prions. Prions are proteins and do not contain nucleic acid. They are synthesized as normal proteins and perform important functions in cells. Thus, the normal form of the human prion protein is involved in the functioning of nerve cell membranes. Sometimes, prions can change the structure. A protein chain arranged in a different way becomes very resistant to any influences that disrupt its structure. In the modified state, it ceases to perform its usual function, and during the meeting with the same normal protein, it initiates the transformation into a prion form. As a result of prions' actions, dangerous diseases affect animals and humans.